Around the turn of the 20th century, scientists using Drosophila melanogaster, the fruit fly, were first able to identify a correlation between the genotype of a fly, that is, whether or not it contained a chromosome that looked like that, the male Y chromosome, or two pairs of X chromosomes, and the sex of the fly. Scientists were first able to observe this because of advances in microscopy techniques that made it able to see chromosomes, and then they noticed that every time they looked at a male fly, they saw this chromosome, the Y chromosome. And that was the first time that there was an indication that something about the chromosome or about DNA might control traits. Decades of experiments that built on each other eventually demonstrated a number of points central to modern genetics. First, that DNA is at least partly responsible for the inheritance of traits. Second, that a molecule of DNA is a major component of what we see under the microscope as a chromosome. And third, that DNA encodes instructions, or genes, for building proteins, the physical substances that both make our bodies and perform the cellular functions that keep us alive. A molecule of DNA is formed by atoms that are held together by many types of forces. A double-stranded molecule of DNA, like you see here, is composed of two strands, one here and one there. First strand, second strand. These are made up of atoms, carbon, hydrogen, oxygen, nitrogen, and phosphorus. These are held together by covalent bonds. As we'll learn later, there are four basic building blocks: the bases adenine, cytosine, thymine, and guanine, A, C, T, and G, that are shown here in their single letter codes. A, C, T, G. These are linked together in a linear chain to produce the DNA strand. Two DNA strands are held together in a double helix structure by weaker hydrogen bonds, shown with these dashed lines running down the middle of the two chains. So, atoms make up nucleotides. Nucleotides are assembled into individual DNA molecules, and two DNA molecules, when hydrogen bonded to each other, make up double-stranded DNA as it is found in a chromosome. An often used analogy for a chromosome is that it is like a chapter in a cookbook. All of our chromosomes together, our genome, is the entire cookbook. By extension of this analogy, every cell in your body is a kitchen. Every cell or kitchen has a copy of the entire cookbook. Only with the entire cookbook, the complete set of chapters, can the kitchen function properly. An individual recipe is, in genetics, what we call a gene. What do these genes do? Each of our genes is a recipe that the cell, or the kitchen, reads to turn basic ingredients into final products. In the kitchen, something like a vegetable casserole. In the cell, genes produce proteins that are part of our cell walls, or enzymes that break down the sugar lactose when we drink milk, or proteins in our eyes that absorb and help us see certain colors of light. Like DNA, a protein is a molecule, a collection of atoms held together by various forces. These are three different representations of the structure of an olfactory receptor protein. These proteins are present in the inner lining of your nose, and when airborne odors stick to this protein, this allows your sense of smell. This protein, like all proteins, is made up of a linear chain of basic building blocks. While the DNA alphabet has 4 nucleotides, A, T, G, and C, Proteins are made up of combinations of the 20 amino acids. So, while atoms can be organized into 4 different nucleotides, which are combined together in a linear string to make a DNA molecule, atoms are also organized into 20 different amino acids, which are combined together, in an order dictated by the gene, in a linear string to make a protein. Other words for a protein that we will sometimes use include polypeptide and enzyme. Like recipes in a cookbook, genes are organized in a linear fashion along chromosomes. Later in this class, we'll learn how proteins in the cell read the alphabet of DNA and how they use the instructions contained in genes to produce more proteins, to carry on the business of the cell and the organism. A general rule, although as always in biology there are plenty of exceptions, is that one gene contains the instructions to make one protein. This is referred to by scientists as the central dogma. One gene makes one protein. More specifically, the central dogma says that DNA is used to produce an intermediate molecule, RNA, whose instructions are used to produce a protein. We will learn about the details of this process as part of this course. Proteins are essentially what every living organism is made of. Much of what you see when you look at yourself in the mirror is protein. So it should not be surprising to you that your DNA is, in large part, responsible for you. How you look, what colors of light you can see, what kinds of food might taste bad to you, the color of your eyes, skin, and hair, whether you have oily or dry earwax, whether you can digest dairy products, and so on. Each of these is a trait. Biologists call traits phenotypes. Although take note that sometimes similarities between organisms can be attributed only to chance, and not to genetics. We often look a great deal like our parents, and we often have medical issues that others we are closely related to also suffer from. What explains these similarities in traits? The answer: similarities in our DNA. At fertilization, when each of us was formed, that initial single cell, the fertilized egg, the one kitchen from which each of us developed, was given a copy of our mom's cookbook and also our dad's cookbook. So many of us look somewhat like one parent and somewhat like the other because we inherited DNA from both. In sum, we inherit our DNA from both of our parents. The central dogma says that our DNA produces proteins, and to a certain extent, those proteins then dictate our phenotypes or traits. This is how genes are related to traits. When you look at our class, there is extensive variation. Why? We're all members of the same species. What accounts for this phenotypic variation? A really big paradox that we'll slowly address across the course of the semester is, any two humans are, on average, 99.9% identical in DNA sequence. That is, there is only one difference in every 1,000 letters when you compare your cookbook to mine. How is it possible that the 0.1% of differences between us produce the quite significant differences that make each of us unique? Somewhere in those very few differences are the changes that make half of us male and the other half female, for example. It's important for us to keep in mind that we don't live in a vacuum. Some traits depend on environment as well, not just on our genes. A big question in modern biology is how to distinguish whether a trait is genetic or environmental or affected by both. To conclude, here are 2 discussion questions that I'd like you to reflect on and be able to discuss in class next time. In addition, here is a list of vocabulary items that were introduced in this chapter.